I think it's time for us to get started again. We have a very, I think, kind of exciting afternoon where we're going to be really looking into some more clinical issues as well with our discussion. I think what Matt Cave is going to present on chronic or environmental liver disease will lead into our discussion about progression of potential environmental progression of diseases as well. I get the pleasure of introducing Matt since he and I collaborate on a couple of studies, both with PCBs especially and Anniston, Alabama cohort that we've both been involved with. But I think we're in for a treat. Matt is actually one of those practicing physician scientists. He still sees patients in the liver transplant unit and so on, both at the VA and at University of Kentucky Medical Center. And he's, I think, going to give us a really exciting talk about environmental liver disease. So, Matt.
Okay, thank you. Really pleased to be here. I'm going to start off with one of my favorite quotes from William Berry. So he's a famous Kentucky farmer, philosopher, environmentalist. And I'm not sure this is his best quote, but I'd encourage you, if you need some good environmental health quotes to read about Wendell. He's still alive, lives about an hour from Louisville. This barn is a picture of my family's farm. My grandparents were farmers. We still own that farm. We still farm it. Environmental health has always been something that's important to us, and even today. So behind the rainbow, or under the rainbow behind the barn, what you can't see is my father's field of Roundup Ready corn back there. So this is something that's always on our mind. So I'm going to start with a patient from my clinic. So this is Paul. Paul's a retired Marine with steatohepatitis and cirrhosis. He used to live in North Carolina and was at Camp Lejeune from 1976 to 78. Presumably he was exposed to VOCs in drinking water. He has traditional risk factors for liver disease, including diabetes and obesity. So interestingly, I also took care of Paul's brother a few years prior. And so Paul's brother had cirrhosis and died of hepatocellular carcinoma. Paul's brother was also exposed to VOCs. He was a chemical worker at Louisville's Rubbertown Chemical Complex and had high-level occupational vinyl chloride exposures. And so Paul's question is, did his exposures contribute to his disease? So objectives of the talk, I want to talk about chronic liver disease and contribution of environmental exposures. And I'm still a relatively young guy, and I wanted to talk to you today about really the impact of some of the training and career development awards from NIH and especially NAEHS really had on my career, because I can tell you right now, I never set out to be an environmental health scientist. I got pulled in from some of these things. And I was recently awarded one of the River Awards, and so I also wanted to talk to this council today about just how important this type of mechanism is to do research on, I guess, personalized medicine, they'll call it in the next talk, chronic diseases, and translational science. And so and along the way, I'll talk about how these things that we do or that I'm doing, we're trying to support the NIEHS mission by increasing fundamental knowledge, translational work, and then training students, bringing in new to EHS investigators along the way. So if you look up, environmental liver disease is not new, and so if you think about it historically, it was linked to poisonings usually. And so we have some aflatoxin experts in the room. So there was turkey X disease is where it was first described. There was Epping jaundice, where I'm from and how I got started in this. In Louisville, there was hepatic hemangiosarcomas in a chemical plant, which you could almost consider a poisoning due to the very high level nature of these exposures. And then there were other poisoning events like in cooking oil, whether it were PCBs or toxic oil in Spain. And so here's as Linda mentioned, I'm clinically active. We still see these hemangiosarcomas from vinyl chloride, and this is one of my clinic patients that had that cancer. So this is what you think of historically as environmental or occupational liver disease. But that's not how my patients come to see me in clinic or in the hospital. They don't come in with rotten corn. They don't say I ate bad cooking oil. So what they come in with is manifestations of cirrhosis, like ascites, which is peritoneal fluid that we have to drain out sometimes, and that's a bottle full of the fluid. Bleeding varices, which is what gets me up at night when I'm on call. So these are varicose veins in the esophagus, which can rupture and bleed, and they'll call me in the middle of the night to get a rubber band gun and place a little rubber band over them. You can see the little blue rubber band there. So I'm 45 years old and still play with rubber band guns. So being a gastroenterologist is nice, but what I want to do is sleep at night. And so if we could figure out what's the environmental contribution to these diseases and do something about it, maybe I don't have to get up and do this. And liver transplantation, multi-system organ failure. So this is how patients actually come in an advanced stage. And so why liver? So why should we study it? And so chronic liver diseases are what I'm going to talk about. And so they're associated with progressive fibrosis and cirrhosis. So cirrhosis of the liver just is a heavily scarred liver. And this has been an increasing problem. So there's a British Medical Journal paper published last year that describes what we knew, that those of us that were clinically already knew, and that there's a horrible epidemic of liver disease. And so this was an epi study. So over the last 20 years, approximately, in the U.S., cirrhosis-related deaths up 65%, and liver cancer-related deaths up 50%. And most solid organ tumor incidents are going down. This one's going up big time. And so, unfortunately, where I live, Kentucky was the absolute worst state for rates of increase. And so this is a big problem. Liver is a target organ for chemical toxicity, possibly due to its role in xenobiotic metabolism. There's a lot of toxicologists in the room that know an awful lot about that. Here on the right is an EPA study just looking at different organs and non-cancer endpoints from various exposures, and liver was the most commonly affected. And 1/3 of the chemicals in the NAEHS pocket guide are known to be hepatotoxic. But yet, hasn't been a lot of research on this, particularly when taking a chronic disease approach. And so, again, patients don't come in saying they've had such and such exposure, usually. But what they come in with is various pathologies. And I've listed some that are liver pathologies. Yes. If Dave's still here, so pathologist is here. So liver pathologies associated with exposures. And so this is kind of the approach of our R35 is chronic diseases. So non-alcoholic fatty liver disease, and then I've listed some types of chemicals that have been associated. Alcoholic liver disease, not as much known. Viral hepatitis, primary biliary cholangitis, which is an autoimmune disease that affects women more than men, which has interestingly been associated with nail polish and UTIs, which women tend to get more than men, and hepatocellular carcinoma and aflatoxin, of course. All right, so what's the value of mentorship and NIH training grants and how these affected my life? So here I am in the middle in 2007 with my little kids. I was just finishing my clinical gastroenterology training. I had a lot of student loan debt from medical school. And considering GI private practice is a quick and easy way to pay that off. But I liked research, and my current boss and long-term mentor, Craig McLean, said, well, why don't you stay in research and we can work with you and try to get some NIH awards, loan repayment program. I had a K-23 through NIAAA, still do a little bit of alcohol stuff, but my passion's environmental health. So, if that was 2007, forward down to 2012, my kids still like to jump off of stuff into lakes, but now they jump off of much higher things. They still play football, but now they play bigger football, and now I've got students of my own. And so this is Van Rita Walling and Josiah Hardesty, who've done a lot of this work that I've shown. And so none of this would have been possible if it wasn't for NIH awards. And I'll talk to you about some things that were particularly instrumental, including a P30 Career Development Award from NIEHS. It made a huge difference in my life. So I've distributed some magazines that talk about the institution where I'm at, University of Louisville, and we've restructured things based on a major philanthropic gift. We have an Environment Institute, which is a new holistic approach that you can read about. And so we have multiple other things, including super funds, some NAEHS training grants, some NIGMS awards for COBRAs, which actually interact because one of them deals with liver toxicology, and this R35 interacts with it too. And so the Environment Institute pioneers a new interdependent vision of health and promotes holistic scholarship. So it's a holistic approach, and so my grant interacts with that. All right, so where do I lie on liver disease in the translational research framework? So liver disease is, relatively under-investigated, and so we don't even know what chemicals cause it and what their mechanisms are, much less how to treat them. And so we're really at that inner ring, and so the approach that we've been taking is to work with epidemiologists to do population studies to try to identify chemicals which may be relevant to human populations, and then develop animal models of those to see if there is an effect, a dose response, identify mechanisms, and then we can go back and verify those mechanisms in people. But we want to get out. I'm a physician. I want to treat patients, and so I want to get out to the outer rings, and I think we're about ready to get there. But anyway, that's kind of our general approach, is identify potential problems with epi and then develop animal models and study those for mechanism. And so we have a very collaborative approach. And so what I tell people, and so this is a great thing about the R35, is I tell people I'm the best collaborator in the world because you work with me, we get preliminary data, you're going to have to be the PI on the R01 that it generates, because I can't be it. And so that works really well. No one's ever told me no that I wanted to work with after I told them that. And so we work with a wide variety of intramural investigators, increasingly big number of academics. We work with EPA and NHRL, especially with Brian Chorley, and here with Linda, and have a new project we'll talk a little bit about with Dale Sandler and her group, and Dave's here from NTP, and Dave really helped me out a lot along the way too, and then Marion Pavick at ATSDR as well. So not as much with industry now, hopefully we'll get there. And so what we're studying mostly is the most common liver disease, and that's fatty liver. And so there's many roads that lead to fatty liver. So this is a common liver pathology. In 1961, it was identified as alcoholic hepatitis. So alcoholic liver disease is a fatty liver. 1981, it was determined that diabetic, overweight people, and at that time it was women, but now we know that men are more susceptible, can get non-alcoholic fatty liver disease. And so in 2010, I described TASH, or TAFL, toxicant-associated fatty liver disease, or toxicant-associated steatohepatitis. So I was, very fortunate for a lot of reasons. So that was the very first paper I ever wrote, described a new disease in my very first paper. So we've been very lucky. But you can see prevalence of this stuff varies. Non-alcoholic fatty liver disease has a 25% global prevalence. And so that's a progressive disease for fat or steatosis. You can get inflammation, that's itis, steatohepatitis, and then cirrhosis, and then some people go on to liver cancer. It's unknown why some people progress, and I think environment plays a role in progression and severity of the chronic liver disease. So what chemicals cause this fatty liver? So, I just described this in 2010, and so not much was known about it, and so I've been accumulating data and a list, and so you can see there's just about any chemical you might work on, at least category: metals, POPs, VOCs, PFASs, pesticides, and over on the right, a lot of these are very high priority chemicals, and so these are very common exposures. And so one of my main messages is, I know most of you are not going to be liver investigators. Many of you have cohorts, but consider looking at liver endpoints in your studies. And so what are the current key hypotheses in the field? And so when I had my first R01 funded through NAEHS, Jerry Heindel was my program officer, and I couldn't figure out why I got linked up with an endocrine disruptor guy. But then it immediately became clear to me, and so Jerry had worked on this metabolism disrupting chemical hypothesis that basically means that chemicals can cause metabolic disease independent of hormone action. And fatty liver is one of those diseases. So non-alcoholic fatty liver disease is believed to be the liver manifestation of metabolic syndrome. And so TASH or TAFLD could thereby be considered to be the liver manifestation of endocrine disruption or metabolism disruption. And of course, there's a lot of overlap, and that's what I've been studying is dietary models. Because people usually aren't exposed to one thing. They may have obesity plus an exposure, and that's relevant. That's what I see in practice. And so that's one hypothesis. And then in liver disease, there's this two-hit hypothesis. So one reason liver transplantation works so great is the liver really doesn't care, if it's treated badly. It can regenerate. It doesn't reject as much after liver transplantation. A dirty little secret about alcoholic liver disease is you feed mice alcohol, they don't get significant liver disease. You have to feed them Libre de Carli diet, which is alcohol plus high fat. And so it takes 2 hits to get bad liver disease in general. And so that's going to be important. Some people call it a double whammy. And so liver is unique in that regard. And so we're looking at a lot of these 2-hit models. All right, so again, I got into this through the long history of angiosarcomas at the Rubbertown, which is our Superfund site, Lee's Lane landfill, which is near Rubbertown. 1942, World War II, U.S. couldn't get natural rubber for military vehicles. They needed it for war effort, made synthetic rubber, made a plant in Louisville, B.F. Goodrich plant. Workers had high-level vinyl chloride exposures. And then hepatic hemangiosarcomas were reported in 1974. We've now had 26 cases from this one plant. The prevalence of this is incidence is probably about 20, 30 cases per year. We've now had about that many from one plant. And so there's Carlo. He was Carlo Tambura recruited to Louisville to lead a health surveillance project. So this was a sentinel event in occupational health. And so it prompted exposure limits for the chemical and then yearly screening and medical exams for the workers. And so Carlo saved a couple tubes of blood and collected all that data. And so this is one of the resources that I walked into when I was a fellow, and how I got started in environmental health was through occupational health. And so Craig McLean asked me to look at some of these angio cases, and while I was doing this, I found these 2 old boxes, wooden boxes that said something about angiosarcoma on them, and I opened them up and they were slides from 1975 from some of the chemical workers that had liver biopsies because the imaging wasn't that good. That was their cancer screening test, was needle liver biopsy, if you can imagine. And so threw one of them up and the liver's full of fat. And well, this guy looks like he's got NASH. And so this was how I discovered TASH, and it was a serendipitous rediscovery because when I snoop around our, biorepository, I find this letter. So this is from Hans Popper, who's arguably the most famous human histopathologist for liver of all time. And so Carlo had sent these same slides to Hans Popper at Mount Sinai. I think this was dated 1979. And so Hans reviewed these slides, and he said on this first one, it shows fatty liver hepatitis, extensive steatosis, focal necrosis, fibrosis. I cannot exclude alcoholic liver injury. If this is firmly excluded, I could consider toxic liver injury, and I hope to receive follow-up information. And so I guess I'd rediscovered something that maybe had been noticed 20, 30 years ago, 40 years ago by a very famous liver pathologist. But they were characteristic lesions of vinyl chloride described, which is what they were looking for. And but what the livers was full of fat. So we studied all these biopsies, and so this is rip-roaring steatohepatitis with the drops or fat droplets. You can see all the blue inflammatory cells. In panel B, this you can see fibrosis and nodules. So this worker had cirrhosis and very high prevalence of these things. So this is actually as high as steatosis that you'll see in bariatric surgery patients, much higher for steatohepatitis and fibrosis, was not explained by obesity or alcohol. And so we call this toxicant-associated steatohepatitis. And so we measured some biomarkers of it. Interestingly, the OSHA guidelines still mandate liver enzyme surveillance for exposed workers, but the liver enzymes were totally normal. And so we've been working on new biomarkers, this keratin 18, which we use a lot now for our epidemiology studies. And so the keratin 18 or cytokeratin 18, CK18, is a cytoskeletal protein that's enriched in hepatocytes. And during cell death, it's a spillage protein, spills out into the blood, and you can measure the whole protein, M65, or if there's apoptosis, there's cleaved by caspase 3, and there's an ELISA for the caspase 3 cleaved fragment. So that's a biomarker of liver apoptosis. And so we use that a lot. There was a lot of inflammation. So the liver drives systemic inflammation, and the most common cause of death in fatty liver patients is cardiovascular event. And so fatty liver is an independent risk factor, partially probably due to increased inflammation. So the liver is the largest solid organ in the body. Not surprisingly, it has the largest reservoir of fixed macrophages in the body that can dump cytokines. And that was the case with these workers, which was not surprising given their liver biopsies. High pro-inflammatory cytokines, low antioxidants, and then disrupted or low adiponectin. And so this looked like an endocrine disruptor too. The insulin was high, the adiponectin was low. And so, we don't do as much as we used to, or probably should, but we still do some specimen bank studies and, use this resource that Carlo left us and apply modern techniques. So we've done some metabolomics in the vinyl chloride workers, and what we found is some more evidence of metabolic disruption, so mitochondrial dysfunction and altered AMPK signaling once you do kind of pathway analysis of blood metabolites. And then we've also been developing animal models. So this is Juliana Beyer, who was one of our liver cobra investigators who helped to develop an animal model of the vinyl chloride exposure. So she gave 0.8 ppm, which is a sub-OSHA exposure limit, kind of an occupational model, 5 days a week, 6 hours a day, and then a variety of diets. High-fat diet and a low-fat diet. And so this is oil red O stain, so liver fat is red. Not surprisingly, you give high-fat diet, you see more fat in the liver, but when you give the vinyl chloride with the high fat, there's more. You give the vinyl by itself, you don't really see much. And so why is that? And so she's shown a variety of mechanisms, including this is seahorse data from mitochondria isolated from the livers of these mice. And she's done time course, which isn't there, but the vinyl chloride causes a depression in mitochondrial oxygen consumption and mitochondrial dysfunction. And so one of the things we think is happening, and you see that even in the low-fat diet, you see it early, but you don't get pathology. We think there's enough mitochondrial reserve until you get challenged with the second hit. And more recently, she's published that she can protect against these effects with ALDO1, which activates aldehyde dehydrogenases. So we think that this VOC is metabolized to a reactive intermediate that's causing some of the toxicity. So why am I telling you all this? Well, so we've been using this knowledge. This is new data from our Superfund. And so we have a cohort. Our theme of our Superfund is VOCs and cardiometabolic syndrome, and we're looking at liver as a feature of that. And so we call this the Health, Environment, and Action in Louisville study. Target enrollment is about 800 or 900. We've got 391 enrolled. This is an interim analysis. And so what we intend on doing is looking at cardiometabolic biomarkers and then planting trees to eat up the VOCs and go back and see if the VOC levels improve and the chronic diseases improve. That's our intervention. And so in this is just our first study, I think, ever using this cohort. And so we measured metabolites of VOCs in the urine. There were 15, and we used this apoptosis blood biomarker. And so the reason that we did this is because a lot of these things are aldehyde, conjugated aldehydes in the urine. And so we're all actually kind of looking at, in a way, aldehyde toxicity in the liver potentially. And so we see for 4 of these biomarkers, there's a positive association, whereas for every increase in exposure quartile, the biomarker goes up, say, 10% there. And so what are these metabolites? Well, they correspond to acrolein, acrylamide, and 1,3-butadiene exposures. So this is very preliminary data. I just got it back last week, but you can see how we've gone from an observation in occupational health, developed animal models suggesting aldehyde toxicity, and now going back to a new human population and measuring biomarkers related to that mechanism. And so that's one of our general approaches. And so again, this is my patient from Camp Lejeune, and so this stuff is very relevant here locally. And so there's trivia question. There's actually federal legislation on environmental exposures and fatty liver. That's the Camp Lejeune legislation. All right, so this is where the P30 was very critical in my career development. So back in 2010 or so, we'd published the first, or writing it up, probably first vinyl chloride exposure study. So the question was, if high-level occupational exposures can cause fatty liver, what about chronic low-level environmental exposures? Can they cause it? And so I was fortunate enough to be able to look at this question through support from P30 Career Development Award. And so what that did was put me and connect me with epidemiologists for the first time. And no one had looked at ALTs and NHANES, believe it or not, until then. And so we did what was the first, wasn't rocket science, but first study looking at ALT and NHANES. And we used the surrogate biomarker, which had been published before, called unexplained ALT elevation, as a fatty liver biomarker. And did a lot of adjustments, a lot of cutoffs. There were about, I think, 200 approximately exposures in this NHANES 2003, 2004. And so what we're looking at is dose-dependent increases in adjusted odds ratios for having unexplained ALT elevation. That means not explained by Hep C, alcohol, iron overload. And so what we found was both dioxin, non-dioxin like PCBs. If you use a different cutoff, you find some insecticides and mercury and lead. And so that was my first environmental health study, again made possible through a P30. And so we've been doing PCBs now for a while, and some of it with Linda. And I don't know why people didn't look more at liver, because I found this MSDS from Monsanto, and so even Monsanto said that the consistent finding in animal studies is that PCBs produce liver injury following prolonged and repeated exposure, and liver injury is produced first and by exposures that are less than those reported to cause cancer in rodents. So liver is the effect that you see first, and I would think if Monsanto reports that this is something that happens that's bad, well, it probably does. So not surprising, but nobody had been looking at it. And so we've continued to look at this through epi approaches. And Heather Clare, who's not in this picture, but Heather was a NAEHS F awardee, and she published this study with Marian Pavic, who's in the center there from ATSDR, and Christina Pinkston, who's one of our statisticians, and this is Anniston 1, which is a cohort of residents from Anniston, Alabama. And so what we knew from some of our animal models that we've done in the interim was that PCB exposures cause hepatic necrosis rather than apoptosis, actually a secondary necrosis. And so we wanted to investigate this cohort because we can use our necrosis biomarkers, and we thought there'd be a high prevalence of liver disease based on the NHANES data, and plus there was a high prevalence of obesity and diabetes, which are the traditional fatty liver risk factors in the cohort. And so we did a cross-sectional study looking at relationships between disease and exposure biomarkers, and we could define a group without liver disease using the cytokeratin 18 biomarker, and then a group with necrotic liver disease and apoptotic liver disease. And And so we wanted to look at associations between these liver diseases and the various PCB exposure biomarkers, which were 35 ortho-substituted congeners. So there's the demographics. Now these cut points for these biomarkers are pretty low, and so there was a very high prevalence of liver disease. There should have been a high prevalence just based on obesity and diabetes. It was 60.2%. These cutoffs these biomarkers aren't FDA cleared, and so we don't know a true reference range. These are probably on the lower end, and so we're kind of maybe sacrificing specificity at the cost of sensitivity. But I thought that was reasonable since this is, in a way, all of this is kind of pilot. But there were some differences with age and sex and race. The subjects that were affected with a necrotic liver disease tended to be older, male, and non-Hispanic white, and that's true for non-alcoholic fatty liver disease in general. And so we found associations, positive associations for the necrotic liver disease category, or the CK18 biomarker, with 15 of the 35 measured PCB congeners. And so, one problem with, so this approach is, and we've done this, since then, is we're adding liver biomarkers to epi cohorts that have already been done. One problem is the reviewers will come back and ask you, well, why don't we want liver biopsy data? Which obviously can't be done and retrospectively, and even if you try to do it prospectively, it can't be done because, one, you'll never get it through an IRB, and two, if you do, that's going to be a barrier to recruitment. No one's going to sign up asymptomatic person for a liver biopsy. And so what we've been trying to address this is a couple ways. And so one way is, if the liver disease biomarker's up, just see if there's associated biomarker changes. Like we know fatty liver will usually have systemic inflammation and insulin resistance. And so we looked at that category of necrotic liver disease, and HOMA-IR and pro-inflammatory cytokines were elevated. And consistent with endocrine disruption, the sum of the PCBs was inversely associated with insulin and leptin. And so we had this necrotic liver disease associated with PCB exposures, pro-inflammatory cytokines, and insulin resistance that we thought increased the likelihood that these observations that we first saw in NAEHS probably were a toxicant-associated steatohepatitis. And so we've been working with EPA and Brian Chorley on other biomarker approaches. So Brian, I think, trained here at NAEHS some time ago as a postdoc. And so he worked with him to measure circulating microRNAs. And the reason we did that is because if you have microRNA expression differences, you can put those in IPA pathway analysis, and it'll actually now predict what the pathology is based on the microRNA changes. And so we measured 68 microRNAs in blood, found some were up, some were down, and then sure enough, when you feed them into the computer, it predicts steatosis, organ injury, inflammation, hepatocellular carcinoma, and so we're applying what they call a liquid liver biopsy approach. So we're working on a variety of approaches here. So this is Banrita Walling, who's an outstanding postdoctoral student, and Banrita came to us from UK Superfund, and she's over her career has developed many of the animal models of fatty liver and PCBs, and she's just a truly exceptional student. Here's an example of one of the models. So we have mice fed a control diet, CD, given PCB 153, which is the most common PCB, high-fat diet, or the combination. And so in this study, you can see that the PCB 153 was really an obesogen, but only in mice that got a high-fat diet. And so the liver slides on the left, they show that the increase in liver fat kind of corresponds to the adipose tissue, visceral adipose in the animal. And so we've come a long way, and I'll show you some of the mechanisms. We've been using these animal models to try to get at mechanism. And so that's really the most, I think, to me, the most impressive thing, and really probably the greatest benefit of this River Award is that I gave a similar talk at Texas A&M last fall, and Steve Safe asked me to write a review article on this. And I was busy, I got stuff to do, but how do you tell Steve no when he asks you to do something on PCBs when you're a young guy? And so I said, well, that's fine. We're just going to, I know all of our data, and we'll just write about it because that's all there is. But I can tell you there's probably 25 to 50 new papers on, and some of you are all centers, on PCBs and fatty liver over the last 6 months. And so many other groups, and some that I've worked with or talked to, are now doing this. And so you can see now that, we've gone from a disease that I wasn't sure existed 10 years ago to first describing in an epi cohort to now many groups around the world are now working on it. And so we're actually finding mechanisms. Some of them are direct liver mechanisms, but some of them are systemic effects interacting with liver. But I think the net effect is the same. And so you get a liver transcriptional reprogramming, and that sensitizes to the effect of the high-fat diet. And so I'm not saying that the exposures in all cases cause the fatty liver disease. What I'm saying is that they attenuate the liver's protective response against overnutrition, so that when you're exposed to overnutrition or diet-induced obesity, the effects are worse. So I think that's an important difference. And I think that's why you see fatty liver prevalence going up, 1 in 4 people, but many of these exposures like PCBs going down. Well, if they're associated, why are they going different directions? And so I'm not saying that the PCBs are causing, I'm just saying that the low dose probably is enough to sensitize. So a lot of that's speculative, but we're getting more and more animal data. And so this was just published this year, and this is an example of why you want a physician working, on your research teams, and in my case, why you want somebody that's actually involved with industry. So sometimes industry, I think it's a bad rap. I've been involved with clinical trials for liver disease, industry-sponsored studies for many years, because when I started, there was no medicines approved for liver disease, and look at what's happened for hepatitis C. So there's no FDA-approved medicine for alcoholic liver disease or non-alcoholic fatty liver disease, but there's many drugs in development because drug companies saw how much Gilead made off of hepatitis C. And so Josiah Hardesty had done hepatic proteomics in one of the PCB animal models, and these are just a brilliant F awardee, and had written a nice manuscript about it, and he'd done this secondary analysis called transcription factor analysis, where you can kind of see, it looks at the protein changes and predicts, what transcription factors activities must have changed to produce those changes. And he wrote a nice manuscript and he gave it to me and looked at it, and I told him, you got to rewrite this whole thing, because buried at the back in an appendix were these lists of transcription factors here that are all down-regulated by PCBs. And to the right, because I do clinical trials, these are the phase 3 drugs in clinical trial to treat fatty liver disease that upregulate these pathways. And so how could a chemical be so diabolical? So like every targeted treatment we have, this chemical is driving things in the opposite direction. And so this is why this has been very perplexing to us, because we look for one mechanism, and it's not one mechanism, it's every mechanism. And so this was a key study for us, and it just came out. And so how could this be possible? And so the next thing that Josiah did was he liked proteomics, and so now he wanted to do phosphoproteomics. And so the reason, and I'll get into some reasons for that a little bit later, but he did phosphoproteomics and found that up to 25% of liver phosphoproteins were downregulated by the exposures. And so just like you can do a transcription factor analysis for proteomics, you can do a kinase analysis for phosphoproteomics and see affected kinases. So high-fat diet, some kinase activities go up, some go down. The Erichlor, which was our PCB exposure, most of them go down. And then look at the interaction between the high-fat diet and the PCB. So the kinase activities are way, way down. And so we kind of concluded here that Erichlor 1260, we call that signaling disrupting chemicals. So there's endocrine disrupting chemicals, metabolism disrupting chemicals, and signaling disrupting chemicals. And so what signal does this disrupt to cause all of these changes? And so we found one, and so this is, I think, a neat story too. And so this is an example of why you want to follow intramural NAEHS research if you're an extramural investigator. And so very fortunate to have Russ Pro that works with me in the lab, and many of you I think know Russ. And Russ retired several years ago, and his wife Betty sent him back to work, and so he was well known to many of you all, and chairman of biochemistry, but he's basically a glorified postdoc in my lab and having a ball. But because Russ is into P450s, he's followed the work of Negishi very well here at NAEHS, and so Dr. Negishi's been working for a long time on finding out the mechanisms of indirect CAR activation by phenobarbital, and a couple of years ago finally figured it out. And so it was antagonism of the epidermal growth factor receptor leading to CAR phosphorylation changes that increased its activity. And because Russ knew that, he said, well, Josiah, why don't you look and see if these phosphoprotein changes you see, because we knew PCBs indirectly activated CAR, at least in mice also, and they do in humans too, but they also can directly. But anyway, because Russ followed the intramural research, Josiah asked that question. The answer is yes. And so Josiah developed this screening assay, medium throughput screening assay using high content imaging, where he treated with various POPs. And so this was a system where you give a radio-labeled epidermal growth factor, and then you just look at how much glows inside or outside the cell, because once it binds the receptor, it's internalized. And so you can treat with antagonists, or chemicals in this case, and see if you block that signal moving from outside the cell to the inside of the cell. And so what he found was that the PCBs, just like the phenobarbital in Negishi's case, inhibited this EGF binding to EGFR. And what was surprising to me is the potency. So some of these are up to femtomolar potency. You can see 10 to the minus 15, so you can do a dose response on these. And it's not just PCBs, it's other POPs. So chlordane was the most potent of all, transnodichlor, and then at a wimpy 10 to the minus 10 EC50 is atrazine. And so I guess it's not surprising to me that pesticides are growth factor inhibitors. Shouldn't be surprising, but many of them are. And so he did some computational modeling, and sure enough, there's high affinity binding sites predicted for PCBs on the epidermal growth factor receptor. And so we haven't published this yet, but I think the proof's in the pudding. Can you overcome this blockade as a therapy? And I'm not suggesting that EGF would be a good therapy for people, but it just demonstrates mechanism. So he's actually able to block the increases in inflammation and fibrosis due to PCB on a high-fat diet by giving epidermal growth factor therapy, because it appears to be maybe a competitive antagonist. Maybe give enough, you can compete it off. So again, not going to go through all these, but these are, mechanisms, some directly in the liver, some outside the liver. The thing I want you to take home here again is that no one even knew that 10 years ago there was any effect of PCBs on fatty liver, and now we're really increasingly dissecting down on the mechanism. For endocrine disruptor people in the room, I think it's important there at the bottom, kind of where it says liver. So the liver is not only a target of endocrine disruption, it also is a cause. So the liver makes hepatokines, which can affect the rest of the body, like TGF-beta, FGF21, and also drive systemic inflammation again. So these are the non-dioxin-like PCBs, and so here's Josiah's drilling it down to kind of a more system toxicology approach, where you see the various receptors and hormones which have identified are abnormal. And so you see all these nuclear receptors down here at the bottom. Most of these are nuclear receptors, not all, but these are the ones that are being targeted for NASH therapy, down at the bottom, a lot of these. And so this is, I think, how we're getting this transcriptional reprogramming that's sensitizing to the effect of the diet. And so where a lot of the breakthroughs are coming, though, and I'm not going to talk a lot about this, is through the dioxins and dioxin-like PCBs, and where there's a ton of great papers that are out this year on the gut-liver axis and how dioxin-like PCBs like 126 change bile acids, microbiome. That's actually a UW work. Short-chain fatty acids, GLP-1, FGF15. And so, again, a real explosion in this area. And so with the dioxins, the liver just doesn't produce stuff. So liver takes stuff in, puts stuff out. It stops making VLDL, stops pumping out gluconeogenesis and glucose, and decreases the FGF21. So, so we're going to talk a little bit about Linda's study, and so using some of that information about the dioxins, dioxins clearly cause, so in difference to the non-dioxin like PCBs, they clearly cause hepatic steatosis, and that's a major project of Tim Zacharewski's lab in the Michigan State Superfund. He's got excellent animal models of it. Criticism of Aniston 1 is that the potent dioxin like PCBs like 126 weren't measured, and nor were the PCDDs or the PCDFs, and so that's why the Recontact Aniston Community Health Survey 2 was done, at least in part, and so fortunately it was pre-designed to have some liver endpoints, and so we've about got these data finalized and ready to submit after it seems like a while. But we ask a different question. So some of the question we asked before, kind of getting at, can the exposures cause a disease? And so now we just want to look at the subjects that have the disease, and are the exposures associated with mechanisms associated with disease progression? And so the exposure biomarker is the total dioxin toxic equivalency here. And then we separated people into groups with liver disease and no liver disease using the keratin 18 biomarker. And then we only analyzed the group that had liver disease. And so we wanted to see if toxic equivalency was associated with a biomarker of cell death, a biomarker of inflammation, fibrosis, and then liver metabolic function with a biomarker of lipid metabolism, glucose metabolism, and protein metabolism. And then it determined adjusted beta coefficients for that. I'm not going to go over these for sake of time. Demographics were similar to Aniston 1. We did have routine liver enzymes on Aniston 2, and they were increased in the group that we thought had liver disease based on keratin 18. And so here's the results. There's an unadjusted model, adjusted without lipids, and adjusted with lipids. It's probably too small to read, but the take-home point was that the exposures were associated with alterations in glucose, lipid, and protein metabolism. VLDL went up. HOMA-B, which is really a marker of pancreatic insulin production, we did B and not IR because we'd seen this before, it goes down. There was no significant effect with TNF-alpha. But importantly, the fibrosis biomarker, hyaluronic acid, although it ends up being weak association in the adjusted model, goes up. And so fibrosis is what kills you when you have liver disease. And so we're getting a clue for the first time that exposure may be associated with fibrosis. And then albumin goes down. Interestingly, though, albumin was predicted in Brian Chorley's model with the microRNA secondary analysis. It said there should be low albumin. It actually was when we did this. So that's ACHS2. I've got one or two more studies to talk about, and I won't spend much time on this, but the PFAS has been a big topic lately, and because of this river, it's really fantastic, because if there's a cohort I want to work with, I don't have to apply for another grant. So PFAS seemed interested, so I called Al Ducketman, who was working at C8 cohort in West Virginia. Said, Al, you guys got 47,000 people. You've published on ALT before, which was positively associated with PFOA and some of the other PFASs that they measured. And so I said, well, we've, doing these cytokines and cytokeratin 18s and finding interesting results in other cohorts. Can we try your cohort? And he said, well, let's try a pilot. So we did 200 of them, and we looked at the liver apoptosis biomarker in blood and a panel of cytokines. And, maybe I'm wrong, but I couldn't find where anyone actually measured cytokines in a PFAS cohort before. So these may be the first and only data. Hopefully someone else has done it. I was kind of surprised, but I just couldn't find it. Maybe I missed it. But it was but it was very interesting finding. So these PFAS are weird for liver disease, and so the apoptosis goes up, but the TNF goes down. So you get cell death without inflammation, at least in this model. And there's some changes in adipokines. And I wasn't really sure about this. And, Angie Slitt at the Rhode Island Superfund's been working on animal models of PFAS and fatty liver disease, because the animal models clearly get it. And she had a poster at one of the meetings once and said, Angie, every liver poster on any fatty liver disease has TNF. You don't have any TNF data on your poster. What happened to the TNF levels in your mice? And she said, well, they went down, but we didn't want to publish it because we didn't believe it. And And I said, well, you need to publish because we're seeing the same things in the human. So it's hepatotoxic and immunotoxic at the same time, I guess. But anyway, these chemicals this was just a pilot study, and I think more work's needed. A little bit on the Gulf study. And so we've been able to do this little pilot with multiple intramural investigators, including Dale and Emily and Larry Engel. This is the Gulf cohort. These samples were taken several years after the Gulf oil spill, Deepwater Horizon, so they're not really believed the levels to be reflective, I don't think, of the actual environmental exposure at the time. But we got them to send about 215 serum samples to us, and we measured that same group of biomarkers before. And so we saw about a 25% prevalence of liver disease. You can see the demographics here. And then we looked for association with exposures. And so we really didn't see much with the VOCs in this study. But what we saw, again, was metals. So that was the very first thing we got out of that NAEHS study was PCBs and metals. It was lead, cadmium, and mercury in that NAEHS study. And here in Gulf, it was lead, cadmium, and mercury again. Some of them associated with cadmium, some associated with lead. So the first one's adjusted beta coefficients at the top, and that picked up cadmium and lead, and that's for having an abnormal M30. And then the necrotic liver disease was mercury, and these are prevalence ratios. I think of the top 75% versus the lowest 25% of having that disease category were adjusted. And they're working on the cytokines now. We measured those. So I'm going to finish up on this because this is a nice segue, I think, for the next talk. And so the approach has been, for the EPI studies was to take, well-characterized environmental exposure cohorts and add liver disease biomarkers to them. But again, in clinic, I don't see patients that say, well, sometimes I do, because people know that I'm into this now, but most of the time people don't come in and say, I was a Deepwater Horizon cleanup worker and I'm worried about my liver now. They come in because they have abnormal liver enzymes and have liver disease. And so we've taken this new approach of so why don't we add exposures to well-characterized liver disease cohorts, not environmental cohorts, just your standard NASH patient that you see in clinic. And let's do exposome analysis in this, and let's see if there's association between exposures and what kills people. And so what kills people is fibrosis, fibrosis, fibrosis. And that's incredibly well known. And the fibrosis even determines the association with cardiovascular disease, liver disease fibrosis, which I don't think anybody understands. But the EPI data clearly show that. And so we worked with Naga Chalasani. So Naga is a very well-established NIDDK investigator at Indiana University, and he had some interest in this, but I pulled him in. We could have recruited these patients from our own clinic, but he had some ready to go, and I wanted to bring in a new to EHS investigator. So he's a big gun in NASH, and but he's new to EHS, and so he had this cohort of NASH patients, and then I love these center collaborations, and so we pulled in Dean Jones and Hercules P30 to do exposome characteristics, and so patients don't get liver biopsies anymore. What they get is FibroScan, and so FibroScan is a vibration-controlled transient elastography. And so that's what we do in clinic for our liver disease patients because people don't like getting stuck with a needle. And so with FibroScan, you get two main measurements. You get liver stiffness measurement, which is a measurement of liver fibrosis. And what the FibroScan does is it's an ultrasound probe with a thumper. And so there's a couple physicians I know in the room. So you may remember you can percuss a liver or you can scratch a liver. And that's because sound or a shear wave travels at a different speed in the liver and other tissues. And if it's a fibrotic liver, it's an even different speed than a non-fibrotic liver. And so what you're basically causing with the thump's a shear wave, and then instead of listening for the sound difference or with the stethoscope a scratch difference, you're actually quantitating it with the sonar. And so you get liver stiffness, which reflects fibrosis, and then when that sonar comes back, it can be muffled. If there's a lot of fat, it can be muffled. and so you get a measurement of steatosis, which muffles the sound. And so we had 150 subjects. Their liver stiffness measurement was 12.2 plus or minus 11. So the cutoff for cirrhosis is 12.5. Probably the more conservative is 14.5. Anything over 20 is bad cirrhosis with portal hypertension. One had a 70. Very unique cohort. The BMI is 36. And the 51% diabetic, a lot of women in here. And the CAP score is how much steatosis. This is moderate to severe. So these are actually sick patients. So we're running low on time, and I'll just finish up real quick. But what we just got these back last week, and so these probably need some work. But what we found was that liver fibrosis was associated with exposures. And I think the most interesting one is going to be this copper benzoate. So it's a copper salt of a food preservative. And I've been drinking Mountain Dews my whole life. And so I learned from Thad yesterday that benzoates in soft drinks. So I don't know if it's the benzoate itself that's associated with fibrosis, or maybe it's just a biomarker for soft drink consumption and high fructose corn syrup. But anyway, 2 pesticides, benzoate, nicotine, and 1 prescription medication. And so they were also able to do some pathway analysis too, because you get the metabolites for free. So you can kind of see how we're kind of changing direction, because we know that the FibroScan is associated with, correlated very nicely with mortality too. So here we're getting harder clinical endpoints. We're learning mechanisms of some of these exposures, like with the PCBs and the nuclear receptors, that, you know, all the drugs in phase 3 clinical trials that are doing the opposite things. We could use potentially those off-the-shelf drugs to actually treat exposed patients. So we're moving from the center ring out over time on translational. So I'll wrap up on that. Started with Paul, who's my patient, doing well, from Camp Lejeune, who had a brother who was a vinyl chloride worker that died of cirrhosis and liver cancer. So, you know, were the exposures, you know, related to his disease? Well, certainly didn't help anything, but for any one patient, it's hard to know. And in clinic, even if I wanted to check, you know, VOCs are transient, but even for PCBs, we can't order a PCB level that's reliable on our clinic patients. So hard to know for any one patient, but again, this river mechanism has been critically important to bring in new people, new collaborators, and move this field, which it was nothing 10 years ago, forward. And I'm really grateful for the training and career development things that you know, I was given through NIH, or else I'd be, you know, doing colonoscopy somewhere. I'd have a lot more money, but I'd be bored out of my mind. So I really appreciate that. I was brought in to NIEHS, and I love it. There's no nicer people in the world than environmental health scientists. And I'm totally convinced of that, because you don't do it for the money, you do it for the love. And I'm right there with you. So thank you, and had a lot of collaborators, a lot of help, and just fantastic students. And so I think this is how my river is going to make the biggest impact, is because we're training students along the way, and we're bringing in new to EHS investigators, because I can't do this work by myself. And I like it better when other people, you know, I'm not competitive with people. If somebody published stuff and beat me, I like it, because that's stuff I don't have to do, because I don't want to do it all. So that's where we're going to have the biggest impact, I think. So sorry I probably went a little long, but I'll take any questions.
Got time for a couple of questions. Terry?
Fabulous talk, man.
Thank you.
Regarding the phosphoproteome, did you guys see any changes in the phosphatases?
Yeah, so for time I flew through that. It was written in the fine print. We did look at several. I don't think we looked at all the phosphatases, which I don't think there's actually that many, but we looked at several of them, and there wasn't any difference.
Okay.
So that's a great question.
Yeah. And then just a quick follow-up regarding sort of clinical management. You know, there's a popularity with diets and changing diets, and the ketogenic diet's quite popular now. And I always wondered about whether or not people who undergo a rapid fat loss from those kinds of diets aren't mobilizing POPs and other things from their fat, and what impact that might have on NASH. Has this been looked at very much?
Yeah, so that's a very real concern, reasonable concern. You know, of course, in bariatric surgery patients which have NASH, they lose weight rapidly, and there are papers looking after post-bariatric surgery where the POPs do go up. There's only one that also looked at NASH, and there was actually, with some of the POPs, there was a surprisingly inverse relationship with I can't remember what component of the NASH it was. So it was a negative association before surgery, which turned positive after surgery when the POPs went up. And so that's just one paper. It's easier, for liver disease, diet's, critical. It's the most important thing. And it's easier to tell people what not to eat than to eat. There is Miriam Voss, who's trained at Louisville, now at Emory, working with Hercules. Just published she works for the NASH Pediatric CRN. She works with them, and so they had a paper in kids with I think it was a low carb type diet, and that actually did well. So the kids lost, even though they ate more calories, they lost more weight and mobilized more fat out of their liver by imaging. So, it was maybe 1 or 2 month study, but it was in a very tightly controlled clinical trial unit. So I think that's probably people always ask about ketogenic diet. There's just not much data. The best data seems to be from the, study in kids. But we always tell them not to drink soft drinks because we get clinic patients that are drinking, at least I always thought I was doing good because these were diet. I said, don't drink 12 regular Mountain Dews a day because some people do. But this has benzoate too.
Okay, so Patrick and then Jose.
Oh, thank you so much. The seamless integration of clinical work with observational and the care for the community, molecular science, really quite beautiful.
Thank you.
I think. But one good way to really rigorously test the hypothesis that PCB is really engaging, surface receptor is use structural biology. Mm-hmm. I'm sure you have been thinking about that. And Linda was talking about, you have Robin Stanley here, right? She taught my biochemistry class at Yale. She's fantastic. So glad to know that she's doing so well. So I'm sure you're thinking about it with your holistic approach. I'm stating the obvious.
Yeah.
I need to say something.
So got to give Russ a hard time. The limitation is, and Terry and I were talking about Russ coming out of retirement. Russ was going to make a recombinant epidermal growth factor receptor, and he had trouble making it. So once you get back into the lab, I guess, and pick up the pipette, things are not as always easy as you remember. But we've actually tried to do it and ran into some technical problems, so maybe we'll try again.
Jose?
Matt, thank you for such a beautiful presentation, a beautiful work. I have 2 questions. In your experience with these clinical cases, when you look at individuals that are being exposed to vinyl chloride or PVCs, for example, it's adiposity or overweight or requirement for developing NASH or NAFLD, or have you seen cases of, relatively speaking, lean individuals with environmental exposure that develop NASH or NAFLD?
So for vinyl chloride, there's probably going to end up being a dose response. So the original cohort from the really old brown boxes that I found, so those subjects had a BMI of like 25.3, probably because they were from the 1970s instead of today, and they were not obese, maybe a little overweight, and so they clearly had biopsy-proven fatty liver, but they were getting probably 1,000 ppm exposures. Mm. They were crawling down in tanks and shipping out plastic that had trapped monomeric gas, and they would go down there, sometimes they would say, in those tanks until they passed out, because vinyl chloride can be used as an anesthetic gas. And so if you're skinny and get that high, you get it. Don't know about the lower doses, at least in the mice. 0.8 ppm, they didn't get any fat in the liver unless they were on the high-fat diet.
So then for the general population that develop these conditions, certainly there's a combination of environmental with some degree of obesity, metabolic syndrome, insulin resistance, whatever you want to call it.
That's what I postulate. That's what I believe. And I don't believe that the chemicals in all cases are causing the disease. I believe that they're making the diet-induced metabolic disease worse.
Yeah, okay. Just one quick follow-up question. Sorry.
It's so much fun.
I know.
We're going to have more fun the next talk, too.
So as it relates to, so you look at all these metabolic pathways, you look at all these key events in lipid homeostasis in liver, you look at VLDL secretion, beta oxidation of fatty acids in liver, and I think I saw in one of your slides adipose tissue. Have you looked at this environmental toxicants or these chemicals, and what changes in adipogenesis, adiposity, and adipose tissue signaling might be affected by these chemical
Yeah.
exposures.
So that's
Now
a,
go-
yeah, so that's a great area. So we routinely do measure DEXA scans and body composition, and then blood leptin and adiponectin. But in terms of molecular biology, the adipose, we haven't done a lot of it. We do have tissues. We always save adipose tissues. If anyone's interested in working on that with us, we're happy to send you samples.
All righty, thank you so much, Matt.
Thank you.
And Matt's going to stick around and participate in our next discussion, too. So we are following up on some topics that we discussed at our February meeting about how environmental exposures may not only be etiologic agents in disease risk, but may also promote or change the severity, et cetera. And Rob and Brad offered to help us lead that. And even so, in your ECB, under the slides, it says Robert Tangway will be part of this, and we welcome you to be part of this, Robert, but the other two guys are the ones who volunteered. So I think they're going to do a little tag team introduction, and then we're going to open it up for more general discussion.
How do I? Oh, it'll be done for me. Okay. I was actually an English major 30 years ago, and so for those of you who are fans of Flannery O'Connor, she actually has a novel, Everything That Rises Must Converge. It's actually a collection of short stories. The metaphor being that precision medicine and exposomics are both rising, and hopefully they'll converge. It's not what the story's about, though. So what is precision medicine? So this is the NRC definition. It's the tailoring of medical treatment to the characteristics of each patient. It's clearly going to be the next wave at NIH and in healthcare. Whether or not it works, there's a lot to be done to really refine it, but it's clearly the direction that medicine is heading in. The idea is to classify patients into subpopulations that differ in their susceptibility to disease, a prognosis, or response to treatment. By doing so, intervention should be concentrated on those who benefit the most, sparing expense and side effects of those who not respond to treatment. And keeping in mind, one of the differences between a medical perspective and a public health perspective, in public health, we're frequently studying healthy populations that might get exposed to something, and what we're really looking at is the probability that some person in that population will get sick. In the medical perspective, the probability that the person is going to be sick is 1. You may not know what they have, but they're there for a reason. So it's a very different perspective. You're not dealing with healthy people, you're dealing with people who have a problem. And that's really, I think, the difference between medicine and public health is actually just the probability of illness is very, very different in the two fields. So what precision medicine really means is understanding an individual's background. That influences disease severity, the progression, and the response to treatment. So what are the most important backgrounds in precision medicine? These are all the potential backgrounds. I would argue that it's actually not genes and it's not environment, but it's actually disease and treatment. It's really how genes and environment interact with the disease in terms of its progression and with the treatment that you actually initiate for the disease. So we have to do studies in precision medicine that will inform physicians on how to better treat patients. And I would argue that we really haven't done a lot of that. I think Matt's studies are a really good example of what could be done, but they're actually relatively rare in the environmental health world and even in the public health world. So the medical perspective is help me diagnose the patient. So epidemiology and toxicology studies actually do help in this regard. If you understand risk factors, you can, physicians use risk factors as a kind of weighted variable in the differential diagnosis. On the other hand, in terms of helping me to treat the patient, epidemiology and toxicology research on risk factors actually doesn't help very much, if at all. In fact, physicians typically are ignoring these results with respect to treatment. It isn't really relevant to what we do. For example, smoking, nobody disputes that it's a cause of lung cancer and heart disease. However, if you show up in an emergency room with myocardial infarction, the fact that you smoked will have no influence on what happens to you next. certainly you'll be told, stop smoking, and you may or may not listen. You're probably more likely to listen. But in terms of the actual treatment, it has almost no bearing. And so we need to do environmental health research that actually looks at the effects of environment on treatment if we're going to actually impact precision medicine. And Ursula K. Le Guin, another author, there are no right answers to the wrong questions. If we want to do environmental precision medicine research, we have to ask the right questions, and the questions, I would argue, actually deal with treatment and disease progression. So this is sort of a schematic of public health research versus environmental research, or excuse me, clinical research. So most public health research is in this time space. So if this is when a disease actually begins to be present, whether you're doing a case control study and going back in time, or whether you're doing a prospective cohort study and going forward in time, you're primarily looking at the time in which the phenotype isn't present, and then looking at when the phenotype eventually is present. If you want to impact treatment instead of prevention, and I'm not suggesting prevention is not important, but if you want to understand how environment interacts with treatment or disease progression, you have to live in this time space. You have to live in the time after the disease occurs, and you need to go forward in time to see how environmental factors influence the progression of the disease or the impact of the treatment. So most precision medicine studies actually need to live in this space, and relatively few environmental health studies actually live in this space. Most of them, I'd say, both basic and clinical or epidemiologic research live in this space. And this is the space that I think we could actually make a huge impact on, particularly in the precision medicine world. So there's a lot of barriers and opportunities if we're going to do this going forward. Clinical diseases can be relatively rare. Liver disease is not super common. You may need networks to get enough patients to establish a cohort. Environment can also be place-based. Air pollution doesn't happen completely randomly, nor does exposure to pesticides or even lead. They happen in particular regions that are geographically variable. However, networks may help with that because you have greater geospatial variability. Other barriers, but also potentially opportunities, that most physicians know almost nothing about environmental health, or epidemiology for that matter. Occupational medicines and the pediatricians and the PACUs are exceptions. They're not the rule. By and large, most physicians actually think genetics is much more important than environment. That's the way they're trained, unfortunately. And they know very little about toxicology as well. And by the other hand, very few environmental epidemiologists actually conduct clinical research. They're mostly interested in causation because it's a public health framework. And for that reason, they don't have easy access to patients. They may know a lot about risk factors, but they may know relatively little about what the treatments are. And so they need to understand what the treatments are if we're going to understand how environment may modify those treatments. So I would argue these partnerships are going to be very, very complementary, that the skill sets of physicians and the skill sets of epidemiologists and clinical researchers actually can work together to do some really interesting research. This is where Brad was going to come in.
So I'd like to thank Linda and Gwen for letting us have a chance to advocate for this. Oops, I already lost my slides. Let me go back. Well, that's not a great start. So Bob and I are from completely different ends of the spectrum. So I'm an adult neurologist, take care of people primarily with neurodegenerative disease. Bob's a pediatrician. And we conceived of this very differently. And when Bob prepared the first round of slides and he sent this to me, I'm like, what's precision medicine have to do with this topic? And after talking to Bob and reading his slides, I became convinced this is actually really a very clever way of approaching this topic. And I think that this is could work really well and be very, very impactful in addressing what I think is an area that's very much underrepresented at NAEHS. So as now this is a year of council meetings, I continue to be struck by how little aging neurodegeneration we see. I mean, I hear lots and lots of child and maternal health, and I think there are a lot of reasons for that. But I think this concept could help address some of what I think is a disparity in addressing really important problems. So I think that this concept of environmental precision medicine works particularly well for neurodegenerative diseases. And in that category, I include things like Alzheimer's, Parkinson's, ALS, and even multiple sclerosis, which has a progressive phase that is considered degenerative. The combined prevalence of these diseases is over 50 million worldwide. If that's not enough to convince you that that matters, several of you in this room will develop one of these diseases in your lifetime. So these are really important diseases that we should care about. I noticed that Kim had a slide about there was a sort of cost to the healthcare system for some child diseases. I looked up during her talk what the costs were for these combined diseases, and the annual cost for Alzheimer's, Parkinson's, and sort of estimating the other diseases is well over $300 billion a year. So these are incredibly costly diseases for which we really need to understand not just disease causation, but how to modify disease courses. And these diseases are Parkinson's is somewhat treatable. These other diseases are essentially untreatable. MS does have some disease-modifying medications. There's variable heritability risk, but large environmental contributions to these diseases. The precision medicine in neurodegenerative disease is focusing really around these very small subpopulations of strongly genetic diseases, leaving aside probably indefinitely or maybe forever the large majority of the cases that have nothing to do with or have very little to do with genetics, and they're certainly not monogenetic. We've had modest success in identifying environmental risk factors for these diseases, and in part because these environmental risk studies are very difficult. It's not like doing these. It's not like you can develop a birth cohort and then study the children until they develop Alzheimer's disease. The exposures of interest may have occurred decades before the disease onset. Diseases like Parkinson's and Alzheimer's have very long prodromal periods. The prodromal periods can be, as I said, can be decades, and so you may be interested in an exposure that occurred in the 20s or teens or even at birth, and you're evaluating somebody at a time when they may be 70 or 80 years old. There's little, and so there's really almost little to nothing known about the role of environment in response to treatment or disease progression. And really, disease progression is most important, the most important variable here. Very large populations of patients nationwide, as I mentioned, and one of the nice things about doing clinical environmental neurodegenerative research in this paradigm of environmental precision medicine is that prodromal period is not an issue when you're looking at response to treatment or progression. One of my primary interests in this topic is that there's dramatic differences in disease progression. So I am a practicing physician, and it never ceases to amaze me. I can see people throughout a clinic with Parkinson's disease. Some can have 25 years of disease course and still work, and others in 10 years are unable to function independently or living in nursing homes. It's nearly completely unexplained. One of the other things about this concept I like is that measuring exposure during the disease course is highly feasible, as opposed to trying to understand exposures occurring prior to disease onset. We can easily build an existing disease cohorts. New disease-based cohorts are also relatively easy to assemble. It's responsive to patients and support organization interest, and I think it's really critical to point this out. So there is. When you're seeing a patient in the office, they will be interested in how they got the disease, but what they want to know is how you're going to make them better, how are you going to modify their disease course. And so if I have the job of selling my research to the patient, saying, well, I'm trying to understand what caused your disease and helping people down in the future, but patients fundamentally want to know what you can do to help them. And this concept of environmental precision medicine, studying the disease course, is really responsive to patients and support organizations, and really allows us to form collaborations that are really impossible when it comes to primary prevention concepts. And as I mentioned, the current genomic precision medicine paradigm is not well suited to neurodegeneration. This does require multidisciplinary teams that typically do not work together, and I've done some of that work myself in my current grants, and I think it's really rewarding when you can assemble those teams. And I think biologic plausibility can be difficult to establish. There is some evidence from Beata Ritz is some evidence of disease-modifying effects of pesticides in her work in Parkinson's disease, and Alison Willis, who when she was working in my lab, did some similar work with metals and survival in Parkinson's disease. But for the most part, looking at the biologic plausibility needed for some of these grants may be challenging. So that's all I wanted to say. We also assembled some questions, and I don't know how you want to proceed with this, Gwen, in terms of the discussion, but our hope was this would be mostly an open discussion.
Yeah. So thank you both very much for starting this off. And so I'll just open it up for comments related to or taking what you've already heard and expanding it and moving along. And you guys can go back to your seats.
Okay,
if you want.
thanks. We wanted to know.
Lynn?
Well, traditional medicine, pre-biomedical science, often used this kind of method to try to help people get better. Spas, TB sanatoriums, sending people to, what did Roosevelt do, right? And so that, this is not, it's not a new idea. It's just that it isn't thought of as a scientific idea. And what I find to be intriguing about what you're bringing forward is that, there may be something to that, from the traditional practice of medicine, just as we've seen many traditional practices, for example, midwifery, that people thought, were superstitions because they just came out of practice and not out of science, that have turned out to actually have empirical basis, but that did not really, didn't come about that way, came about through practice. So I think it's kind of an intriguing idea. As a pediatrician, there's an obvious example, which is asthma, childhood asthma, where environmental interventions absolutely can improve the course of the disease. But we understand that, so maybe we don't think of it as that. Maybe we think of that as something else. But I think people understood that before, but now we can see the basis for it. And I think what's been important with that has been being able to connect the environmental intervention to the actual pathophysiology of what triggers the symptoms, which I'm not sure that we really understand, with the chronic neurological diseases that affect the elderly. The other thing that kind of pops into my mind is that, I'm going to say something that you probably won't agree with, but that at least to some of us as epidemiologists, some of the variability in course of some of these diseases like Parkinson's and dementia, some of us haven't been convinced that it's all actually one diagnosis. That that the variability could be coming about because of having different pathologies involved, and I think that would be an extremely important piece. I think, is whether they're cohorts or there are their patient already pre-existing clinical studies where people have been much more careful about delineating pathologies, for example, involved with dementia, to be able to kind of slice and dice to make sure that that's the variability, that the variability you're looking at is the environmental intervention and not the actual disease.
Yeah, and this discussion obviously isn't all about neurodegeneration. It just happens to be one of the things I really thought was where it was relevant. Just to respond, that's not what this discussion is about, but just to counter slightly. The reality is even in the best cases, if you take well-characterized cohorts that that come to neuropathology, the final pathologic diagnosis, despite the common pathologic diagnosis, the disease course can still be quite variable. Extremely variable. That doesn't mean that we shouldn't be. I'm a lumper, not a splitter, but I lump because I don't have evidence to split. Once you give me evidence to split, then I'll be happy to do so. So your point's well taken. We just haven't been smart enough to figure out how to split in a logical way, and that's been a real. It's been a challenge in neurodegeneration in particular. Not as much in other diseases, like neuromuscular diseases have been very easy to split into 1,000 different pieces. Right, exactly.
So, and one of the things that maybe didn't come across in the presentation is, there have been enormous advances in exposure science in the last 10 to 15 years. We can measure, in panels, we can measure hundreds of chemicals now. That wasn't the case 10, 15 years ago. Certainly wasn't the case 50 years ago. We have untargeted assays where we can find unknowns that may actually be related, that we can dig deeper into or try to figure out whether or not they're actually the cause of a treatment failure or more rapid progression. We have geospatial technology and big data technology that also didn't exist even 5 years ago. So much as genomics has reached a point where it can start thinking about this idea of precision medicine, I would argue that environmental health and exosomics is getting closer every day, and there's a lot we can do. And that it wasn't even possible 5 years ago. And I think that's another reason to do this. And also, it's hard for me to conceive that whatever led to your type 2 diabetes, and even if it's a subset of type 2 diabetes, if phthalates are actually obesogenic, if they actually induce insulin resistance, that you wouldn't be more susceptible, and that therefore, if you have an unknown exposure to phthalates, there may actually be some clinical advice that can be given that might improve your course. So I think it may actually, the patients may actually be the largest group of susceptible people to the environment.
So I feel that the precision medicines actually is getting closer and closer because, see, we're talking about, Bob, I really like your presentation about the 2 time frame that disease occurring and also prior to that disease. So you actually you actually lay out a 3rd dimension of time, and there are multiple variables, but with the artificial intelligent learning process, it's truly possible to integrate all these data points and in 4th dimensions from maybe day 1 of birth or even prior to that, and then be able to actually look at how the disease progression starts way before one can diagnose that, right? And then actually predict it, and then follow its course down the road and be able to design treatment. So I think the opportunities is immense right now, and we just have to really entice those people who make trillions of dollars doing other professions to come into this particular field and helping to do some of these. If they can move all the goods around the world and all the wealth around the world, they can actually move this mountain very easily. All the barriers that you lay out will no longer be barriers.
Taking as a starting point that I think this is a really good idea for all the reasons you mentioned. It's a great opportunity, I think, for research, for generating knowledge, and for the Institute. So overall, I support it, and I'm thinking now, how do we get there? And I want to encourage the inclusion of all affected populations, certainly the disease interest groups, but also those who would be involved in exposure reduction and in development of this research agenda. Because we need to think about not only the clinical implications that this research will have, but also the novel therapeutic recommendations, actions, resources, practices, and policy changes it's likely to recommend. So it's quite possible that equity-focused stakeholders will have unique questions and concerns we may not have thought of, even ranging from the name. Because I have to say, on first blush, the term precision medicine has the ring of treatments available to the privileged few, maybe ranging to the kinds of diseases and problems that should be prioritized. So I think it's really important that we think through, at the same time that we're thinking about the knowledge we can gain about how to co-develop systems for exposure assessment, management, payment, and policy at the same time. So, for example, if we find that diabetics should not be exposed to phthalates, how can we provide phthalate-free environments? How can we leverage this to prevention policy? How can we assure that everyone has equal ability to benefit from this knowledge? If we are developing precision medicine that results in medical treatments and for drug development, right, that will eventually be covered by health insurance, but environmental treatment is not. For example, finding that indoor air quality is critical for COPD patients is really important. Figuring out how to pay for filtration systems, never mind their utility bills, is the equivalent of drug development. But it requires a totally different way to think about clinical translation, therapeutics, dissemination, and implementation. So my basic idea is I think that it's really important to have a very inclusive process for developing this research agenda.
Maureen, and then Brad.
Could you really about me a way to say this specifically? I was thinking how to articulate this. I support it. This is a really great effort and a new direction for us. But I'm also aware of the ethical consequences of this. And so how do we, ethically, from a patient perspective, can we do something about what we find clinically? Because otherwise, it's not very helpful to a patient to say, oh, and I also know your Alzheimer was caused by this, and we're going to just watch you, progress. So can we do something about it? And then secondly, there is, in addition or parallel to precision medicine, we're also increasingly talking about precision public health, because some of the individual exposures, and although it's a clinically focused on one patient, are actually community-driven and community-based and contextually influenced by communities. And so if we even can combine those two domains, it might be even more powerful.
I can, to answer the first question, I think because we're not talking about what caused your Alzheimer's disease or what caused your diabetes or what caused your autism, we're talking about something that's affecting the progression. I would argue that there is something we can offer, because I once took care of a patient with autism who had pica, which is very, very common, and was eating newspaper, which had contaminated with lead ink. And when we discovered that, some of his behaviors that he presented with were headbanging, which were new and greater onset, when we discovered that, we were able to do an intervention that reduced his blood lead. And also, this was in the 1990s. Nobody reads newspapers anymore, so this probably won't happen. But there was something we can do. And I think in this paradigm, actually, we're more likely to offer something that will actually improve their course. It won't necessarily treat their disease, but it'll keep their progression from getting worse or bring it back to their original baseline. And I think that's much more likely in this paradigm than when we're doing disease causation research. What was the second part of your question? I don't think they're mutually exclusive. Obviously, I think precision medicine's easier because it's at the individual level. Precision public health, because it's at the population, is a lot more difficult. It's more difficult. Public health interventions have always been more difficult than medical interventions. Healthy people have less incentive to change, if they perceive they're healthy. No matter, even if they have bad health habits, but they don't have a disease, they're less likely to change. People who have a disease are far more likely to change. And I would actually argue that if we're interested in getting things like phthalates out of products or what we think are toxic chemicals out of products, getting disease advocacy groups on our side is actually going to be very, very powerful.
Okay, Brad, then Matt, then Lynn, and then Jay.
I'm going to respond to both of those, just building on what Bob said. In terms of disease progression, right now we have nothing that affects the disease course for Alzheimer's, Parkinson's, or ALS. Nothing. So there are no disease-modifying therapies. So, and the environmental world we've sat on the sideline where NINDS and NIA have taken a lead in trying to develop disease-modifying therapies, failed miserably at every level. I think it's a really great time for us to try to get involved in it. And I agree, it could be very challenging to implement findings, but right now, since we have nothing, I think we would do a great service to our patients if we could identify factors that actually could be modifiable, because right now we don't. To get to to respond to Katrina, the precision medicine concept that you commented on, where only the wealthiest benefit is the current model, right? So you're only going to be in a trial for LRRK2 Parkinson's disease if you have enough money to see a neurologist who knows how to get the DNA test and you're gene positive. In this model, you're actually paying attention to the poorest of the poor because they're the ones that are most affected by many of these exposures. So in many ways, this is a more inclusive disease model than the model that we're using currently.
Matt?
Yeah, so this on?
It's on, yeah.
Okay. So you could probably tell through my talk, I was kind of independently moving in some of these directions too. And I think you can probably, as an institute, NAEHS could get a lot of bang from the buck from utilizing clinical trial biorepositories So, for example, NIDDK may have a ulcerative colitis clinical trial and a clinical research network. And so the benefit of clinical trials is that you have longitudinal follow-up and you have placebo arms. And so in the placebo arm, you can get learn the relationship between an exposure and natural history. And then in the treatment, you can look and see if environmental factors modified response to therapy. And so not only would the other institutes be a resource through their big CRNs, don't forget about industry. So again, I do a lot of industry-sponsored clinical trials, and they biobank. Every study has a waiver for biobank for future research. And so drug companies have tons of these cohorts too, and so it'd be relatively inexpensive just to add exposomics to some of these placebo-controlled clinical trials and learn a lot about disease progression and response to therapy and leverage resources of multiple institutes as well as industry. And one other quick comment, about community. So if we're talking about patients now, there's other definitions of community. So there's patient advocacy groups for any disease that you have. So I mentioned the rare disease, often in women, primary biliary cholangitis. So they've got a great advocacy and support group. And so you can almost consider them a community, the community of people that have the disease, because there are a lot of advocates for that too. So a community based on whether or not you have a disease and not what your zip code is.
Yep, thank you. Lynn was next, and then Jan.
Yeah, a couple of other ideas. One is that there are actually patient cohorts that have been established, and most of the cancer centers have them, for example. And the NIH actually supports a number of them in other institutes. And so actually, people with diagnoses can be studied over time longitudinally. And it is possible to, as you can, with children with asthma, to look at natural experiments, such as that people who happen to have these diagnoses happen to live in different environments, and to see if their course is altered by living in different environments, not because we've put them there. So that's something that could be done. And I like the idea of trying to somehow incorporate some of this into clinical trials. I was thinking about some chronic diseases, and actually, to me, the liver diseases are like this. So I thought maybe you'd address this, which is that there are diseases like cancer, where you've been smoking, you get the cancer, then you have the cancer, so then you have cancer, right? But there are other chronic diseases where the exposures that cause the disease can continue to make you worse. And I think of liver disease as that way, and I think also diabetes is kind of like that. A lot of metabolic diseases are kind of like that. And that might be another path to take, and that is those kinds of diseases where we think there's a reason to assume that some of the causal factors also can be contributing to continue to make the disease worse, or the removal of those might make you better. The final idea that I wanted to put on the table is that there's been a lot of thought about the environments where people receive medical treatment and the extent to which those environments might be able to be more beneficial to health. And like hospital architecture, trying to improve that, bring nature in, fresh air, all of that. And that, I think, is also amenable to study, to more rigorous study. And it could be that we're underinvesting in that, overinvesting. There might be people in the medical care industry who could be motivated to help to study that kind of thing.
Thanks, Jan.
Yeah, first of all, I'd like to thank Brad and Bob for bringing this into discussion. I think it's an incredibly important area, and I think it's an area that there really is the opportunity to get a tremendous amount of traction. I don't think we can underestimate the ignorance of the physician community about environmental health. I mean, I think, those of us that are in that community know that we didn't learn any of it. Most people aren't interested in it. I came to environmental health quite late in my career, knew nothing much about it before. And I think that it is important. I think part of what's been difficult is that so much of our data has either come from animal models or from epidemiologic studies. And especially in terms of epidemiologic studies, so much of what I think the physician community sees in terms of the solutions to some of that relates to public health, relates to policy, relates to things that we don't have any control over for our patients. And so I think that bringing the physician community into a role where they do feel as though they have an ability to impact is really going to be an incredibly powerful thing to do. And so I really want to, I just want to applaud you for bringing that to the fore, for all of the reasons that Brad has outlined in terms of the opportunities, not just in pregnancy exposures, but in other exposures and ways to think about doing studies beyond that, I think are really important. We know that patients are very motivated. They're much more motivated than people that don't know that they have anything. And so again, this is a population that will allow us to get more public opinion behind us as well. So those are my personal comments. The other comment I wanted to make is one kind of related more to a programmatic perspective that Linda also wanted me to bring forward. And that relates to the All of Us cohort, which was initially called the Precision Medicine Initiative, but given that we don't know what precision medicine is, the name was changed. So as many of you know, a lot of money is going into developing a very large study that is going to be collecting a lot of different kinds of information of, 1 million people across the country in different settings. This has now been quite nicely rolled out. A lot of people have been recruited to, I wish I had the numbers for you, but I don't. I'm sorry?
Around 200,000 are already enrolled. And
Yeah, probably more than that, though, at this point. It's really pretty high.
200,000.
Pardon?
200,000.
Two-
plus on it.
oh,
200,000 plus or
Oh,
minus 50.
I sorry.
200,000.
I didn't,
And it's only a year.
yeah, I didn't hear you say 200. That's what it was the 100 part that I missed. So now we're good. And there's a lot of very interesting infrastructure that is going on around this that relates to consenting and relates to biobanking and relates to sort of artificial intelligence in terms of bringing things together. So I think the resources of the program for our extramural community in particular are really huge. And it's something, it's not a perfect study. It's not a beautiful epidemiologically defined group. And so, we need to get away from thinking about it that way. But having said that, there's a lot of resources that are going into this and a lot of data that's going to be collected. A number of the institutes have started thinking very seriously about how to potentially roll All of Us or some of the resources from All of Us into their extramural program and to think about how do you design studies that are really designed to address the goals of your institute within the context of All of Us. I think we haven't started doing that to too much extent at this point, but it's something we may want to be thinking about. There's one other point I was going to make. Oh, yes, from the perspective of environmental work at the moment, I think that all of you would be incredibly disappointed by what is being collected. It's just absolutely minimal questionnaire type stuff, even though it's supposed to have a major environmental focus. So again, the more push that can come from the extramural community, the better. And hopefully there will be things that can be added on as sub-studies, and I think that's probably where the biggest power is going to come from the environmental community.
Gary, and then Brad.
Hi. I've really enjoyed this discussion. I just wanted to bring your attention to survivors cohorts that NCI supports, and there are several, and some have banked biospecimens with many years of follow-up. And I'm not aware of any work that looks at environmental exposures and cancer progression and impact on treatment. I think that's a real opportunity. I did pull one study that looked at heavy metals and suggest that it stimulated breast cancer progression, which also decreases sensitivity to treatment. And I think that our cohorts could address some of that. But even with the risk cohorts, I struggle with that because often we design these cohort studies and folks are older and we measure exposures at baseline. And I think that the relevant exposure period may have passed. And so are we really measuring risk or progression? So the main thing I wanted to point out is that we do at NCI support resources or cohorts that would be amenable to looking at some of the things you discussed.
Brad.
So just responding to just want to comment about the All of Us cohort. I don't know that much about it. We did talk a little bit about that in our initial discussions, and I think one of the challenges there is it will work really quite well probably for some more common diseases. But if you have a disease like ALS, for example, you may have like 20 cases. So it's really limited. But there are plenty of ways to get at those kinds of populations for the rare diseases. And in terms of the challenge, I mean, just to give you an anecdote of how challenging this can be, I was part of a big NAEHS 10-year clinical trial for a bunch of failed neuroprotective drugs for Parkinson's disease. So in Parkinson's neuroprotective trials, you not only have the placebo group, you also have the active treated group because they behave the same, right? Because they're not getting any better. So you got the entire population. It could be your study population if you have an environmental variable. And I simply tried to add to this data set a geocoded address for every person, and it was blocked at 100 different levels. And it was 10 years of longitudinal data that we could have had on these people, knowing where they lived, and it was very, very meticulously phenotyped. 1,000 people with Parkinson's disease would have been a great opportunity to just study residential history, and even that was challenging at the very simplest piece of data. So these are, we have barriers that are just sort of artificial and unnecessary in some of these studies.
Matt, standing up again at the mic, and then we can go back to the table.
Fall in the rabbit hole of naming all possible cohorts or possible partners, but I do think there's one worth mentioning, and the All of Us cohort made me think of this. So the Million Veteran Project would be a great resource. So no one's mentioned gene by environment interactions, which is what I immediately think of, in personalized medicine. And I understand they're genotyping 1 million veterans, and they're not doing any exposure assessment that I know of. So if you can add that, no matter what disease you want to study, 1 million people, if you had all the exposures and all the genotyping, you could probably make progress. So just don't forget about the VA.
Yeah, the All of Us obviously is all being genotyped.
Shikme?
Okay, so I want us to keep thinking outside the box, okay? So basically, the VA has a registry for burn pit exposure. People who register is already over 20,000. They can just register that, and they can volunteer to actually participate in a study. So that's point number one. Point number two is don't forget about consumers' force. So just like the Susan Coleman's entire cohort is actually based on people volunteering. So we have all these people doing 23andMe and other DNA testing. Well, what prevents us from actually asking for their volunteership? Okay, so this is a really great community exercise, and everybody gives their loved ones a gift on Christmas for a DNA testing. So this is consumer forces, and the age, what is it? AARP. AARP is a huge organization, and why can't we use some of those as a vehicle? And when we worry about all this geospatial tracking, my son has given up his privacy because he said that it's better for Google to manage his life. So basically, what I'm asking is we got to think outside the box, and in order to elevate, and the precision medicine is actually broader than just exposure, lifestyles, zip codes, and all that. It's just a tremendous amount of data that actually makes why we single personally is unique and our disease is unique.
Yeah, go ahead.
So, Shukmei, you made a really interesting comment, and it's making me think a little differently, too. We had a whole discussion this morning about the need for communication and how it's done and the training in that. And I think one of the biggest barriers we potentially face in the environmental health community is the lack of recognition that environment really impacts our health. I think when you deal with the medical community, that is the overwhelming message. And we hear that again and again, where all these cohorts are doing lots of genotyping and essentially no environmental stuff. So I think that we need to be thinking about a communications campaign.
So this brings up a very important point. We always talk about environmental toxicant pollutants and all that. Those are words that people don't like to listen to. All this, so what we classify as like whatever is in the drinks, the soda drinks, we can consider as a consumer product. We can consider as a lifestyle. We can consider as a food choice. So in that setting, the communication is so much easier for people to accept and participate. And we very soon we will have a lot of, I think we don't want to feel like we're being studied. We want to participate. So if we can turn the table as being studied into participation, I think it will be possible.
Okay, so that's what's happening in the All of Us cohorts. We have participants in the study. They're not considered subjects, they're participants. But I really, and I think what you just brought back, what I was trying to get to was the point that communication, it has to be not only message delivered, but message received, which means you got to know how to deliver something that people will accept and that people will take advantage of. I think some of the things that Brad said related to, dealing, and several other people, whether it's advocacy groups or disease cohorts or people who are already in clinical trials, I think those are populations that are potentially, that we may have the opportunity to take advantage of, to ask the questions about the exposure that occurs in them, and then the idea, if you've got a clinical trial that's being followed for years and you may have samples where you can find out, well, what was the exposure at time 0 and then at time 3 years and then at time 6 years and then at time 9 years. And by 9 years, you know what's happened to those people. And some of the people may be doing really great and others nothing, has happened. So I think that these are really exciting ways to begin to think and approach it. I have to thank my colleague Matt again for kind of giving us an example of how that has been or can be used. And Matt got into the environmental sciences field because he had a mentor who said, hey, this is a great opportunity. We need to be working to develop more mentors who will say, hey, this is something that you can do. Obviously, I'm in favor of this idea.
Are we out of ideas? So, I told you yesterday that I had gone to this workshop last week about reduction of personal air pollution. And, it's that same kind of thing, like the practicing cardiologists who were the clinical trialists, they invited ones who had open minds to this concept. They didn't know much about the literature about air pollution, so they were convinced that it was possible to do. And, that kind of question about what's the universe of clinical trials where, so characterizing the phenotypes or getting the clinical information is really expensive and laborious and all of that. So if you can capitalize on programs where you already have that, then we can add the other really expensive part, which is the collection of the environmental data. But, you could do that in a selected, targeted way maybe what, we need is some proof of principle cases as examples and then sort of shop them around so that clinical audiences understand and appreciate this.
You could start off actually with pulling some of the existing things that we already know. So that, for example, with asthma, we do know about certain things that will progress the
Exacerbations
or exacerbate
some
stuff, and then things
symptoms.
that if you remove them will help prevent. And I think that we know some other things related to air pollution and lung health that we can also look at. And I think that there are some other areas that we can think of more off the top of my head. But I think that, liver is something where we may be able to look at that too.
I think we've had a few isolated cases of trying this in our portfolio over the years. I know in a Long Island breast cancer study, after the case control study was done, there was a follow-up for survivorship for the cases, and they looked at PCBs, which was thought to be an etiologic agent, to see if that differentiated survival. So that's just a concept out there. It could be expanded within the cancer world, and chemicals that are thought to be, they may not be etiologic agents at all, but they may, work after the disease is here. So, you have to switch your mind there. What would you be looking at? We had a grant years ago about autism in a children's center that they looked at children with autism, and they were interested in hand-to-mouth behaviors to see if maybe lead on dust and other exposures, might be related to the behavioral severities of some of the phenotypes of the children with autism. So you can see, these didn't pan out to sort of be what everybody would have hoped they would, so it sort of died off. But maybe today we have more ways to do things and measure and to use technologies to be able to characterize both the exposure and the phenotypes. And maybe it's just time to try again.
One of the things that struck me is when we were putting this talk together, and even when Brad and I were having exchanges, it's often hard when you have a public health perspective to not think of the cause. And I think it's important to emphasize that it doesn't, we're not saying that such and such caused your disease. We're saying it's making it worse or keeping you from getting better in your treatment, which is a different message. And I find myself reverting back to cause a lot. And I have to remind myself not to do that. And I think that has to be part of the message. So it's not, so people don't think we're doing the same thing we've always done. This is something different.
Andy.
Just to follow up on your comments. So several years ago, we tried to develop a environmental exposure sheet for our gene banks, right? A GREE collection, but it really ended up not going anywhere. And I think it's probably because of our lack of understanding of potential value. But now we have a 10,000 whole genome cohort, sequence cohort, and I would love to see whether we can explore adding an environmental questionnaire on them.
We might not want to only think of exposures in the external sense. We might want to think of microbiome. And as we know that now, as we're beginning to appreciate its key role in many health conditions, and we talk about autism, there are the papers that just came out recently, demonstrating in a small number of patients that putting in a healthy microbiome reduced the symptomology of the children with autism, and there are animal studies that support that. So that, we might want to think not only what is external to us, but what's in us.
If I can add to that, and it brings me back to one of the things that Shook May said, I think in the clinical context, it can be important to figure out ways to message these things as positives. The absence of the negative, the bad the good microbiome, not focus on the bad microbiome, but the good microbiome, the positive environment, the better nutrition that enhances your health. Those are the things I think that, you know, many people are looking for when they have a diagnosis and they're trying to get better. And I think it's less off-putting in terms of thinking about the environment to think about it that way.
Lynn, I get so frustrated with myself when I'm talking about some of the environmental contaminants and all the terrible things that they do. And like my kids who are doctors will say to me, well, what's the good news? What else can we talk about? And I think by kind of inverting it, and maybe by taking this issue of understanding what may make your disease worse and we can therefore remove it, that would be an entirely different message. So that's the communication again.
So, you know, when you look at reducing asthma exacerbations and putting the positive spin on less ER visits or, you know, less school days lost, I mean, those are all positive statements, and healthy environments for children that reduce days of being sick or time taken off from work, so taking care of the kids. I mean, I think if we picked a few case studies, we could create those messages around, look for data that would support those kinds of things. That would be quite interesting to then, you know, put those on our banner, on our website.
But I want to go back to the issue of the chronic diseases, not just things, I mean, asthma is chronic, but maybe some of the neurodegenerative or other more age-associated, because when you look at the cost to our society, what is 50% of medical costs are associated with the last 6 months of life? You know, think what could happen if we could understand that the progression of Alzheimer's disease is related or could be by removing certain kinds of exposures, internal or external, you would slow down that progression. I mean, you can just think of the tremendous benefit to the population.
All right, who wants to have some closing comments on this? You all look tired. So, Sue, yeah, it's been great.
I don't have anything specific to say. I just want to say I think it's been a really useful conversation, and it's a really important topic, and we should keep this going for sure and think about it more and figure out how to keep it moving and actually get some good research started. I think it'd be really exciting. And, you know, Bob, as I'm listening to you, I'm thinking, yes, it's hard to sort of reset our minds to think about, we're not thinking about what causes this. We want to think about, you know, how can we slow the progression, or how can we, you know, improve life for people who have already been diagnosed, or how can we make sure the treatment's effective? Those are really important, interesting questions that we haven't been thinking about very much. So
yeah,
I just want to say I think this was a great conversation, and it's a really important topic.
I think that I don't know when you'll see some kind of concept on this, but I think it's highly likely that something will follow. I just want to make I want to make one comment that hopefully we won't have bad weather in September, so I'm hoping that everybody will be able to come to my house for dinner that night and we won't have to cancel. I had to cancel 3 dinners in a row. We had flooding, we had ice, we had something else. Anyway, hopefully September we won't have any of those things.
Yeah. Well, thank you very much for taking the time to think this through and leading the discussion, and for everybody's participation in it. Before we end the meeting, I just want to thank our staff, who I think did amazing work for this meeting. Presentations were awesome, and your engagement in discussing deeply all of those concepts is really helpful to us. You know, we still have a lot of work to do on those projects, and we take all of your comments quite seriously. Thank you to Liz for getting us through another council, and for Pat for all of the coordination efforts. And now I'll turn it over to Linda.
I thought I already had the last word, so, oops, right? Everybody, travel safe.
Safe travels, yeah.